So let's finish our or continue our discussion on cell division, and this time we're going to talk about mitosis. So before we get started, we have to know there are three different types of animal cells: somatic, germ, and gamete. In a somatic cell, these are your body cells. Okay, so if I say, hey, name a body cell, you can say something like a skin cell, a red blood cell, a bone cell, a muscle cell. Those are all body cells. These cells have two sets of chromosomes. Okay, so in humans, that means they have 46 total. Okay, and that would mean 23 in a set. Now, they usually undergo mitosis to make more copies of themselves. Then you have what's called a germ cell. And now, this is not bacteria, viruses thing, okay? This is just called a germ cell. These are cells in your ovaries and testes that become your gametes, okay? So they are called spermatogonium and oogonium. And those are the cells that will divide to make the sperm and egg. They also have two sets. Now, to make the sperm or egg cells, they undergo meiosis. But to make more copies of themselves, they will have to undergo mitosis. Lastly, we have the gametes. These are your sex cells, okay? These are your sperm and egg cells. These cells are unique in that they have one set. They have 23 total chromosomes. They do not undergo cell division, okay? They are going to unite to make the zygote, which becomes an organism, okay? So, somatic cells, red blood cells, nerve cells, bone cells, muscle cells. You see the nucleus in these cells, you know that these are eukaryotic cells. Germ cells, spermatogonium, oogonium. They give rise to your sperm and your eggs. So through a meiotic division, they make sperm and eggs. And then you have the sperm and the egg cells, which are your gametes. Here's the karyotypes. When we talk about sex, you see that there are 23 different types of chromosomes here. 1 through 22 are called autosomes, and then you have your sex chromosomes, okay? So 1 through 23 are autosomes. And then these are your sex chromosomes. This is called a karyotype. So for any given chromosome, you have one from mom and one from dad. And that's a set. So you get one set of 23 from your mom and one set of 23 from your dad. And now, I want you to think, what do you know about mitosis? So what can you tell me about mitosis? And it's a type of cell division. We should know that, yes. We know from the other slide that somatic cells do it, or use it, and to a degree, germ cells. We know this is a form of asexual reproduction, and you produce two identical cells. Okay? The purpose is for growth and development. So if you damage your cells or you're growing and you need more cells, you need to be able to make copies. So let's look at how this is achieved. So here's mitosis. Okay, you'll see you start with one cell, you end with two. What I will note here is that this cell can re-enter. Okay, so one thing you should see that this is a cycle that can continue as long as you need to make new cells. You'll also notice that it's broken down into these different phases, okay? So there are phases that make up mitosis, and in general, it's usually called IPMAT, and sometimes it might be 2Ps, okay? Or PPMAT, because this I is debatable if you include it in mitosis or not. So let's look at what's happening. So usually in the I phase, would be here. And this is interphase. And this is where the cell grows and the chromosomes are replicated. Okay, so this is largely signified by growth and DNA replication. Then once you enter prophase, you start to lose this nuclear membrane. So this has to break down. The spindle fiber forms. Okay, so you're going to duplicate the centrioles, and then they start to move to opposite sides, and you form these spindle fibers. Your chromosomes condense. Your DNA condenses to go from this chromatin you see here into the chromosomes that you're used to. Next, sometimes people call, so here you see it's called early and late. Sometimes this is called prometaphase. So here the nuclear membrane goes ahead and completely disappears. These spindle fibers are going to attach to the chromosome so that it can start to move them around. In metaphase, this is metaphase, they line up in the middle. Okay, so metaphase is characterized by these chromosomes, a single file line in the middle. Then you get to anaphase, and those chromosomes come apart. Okay, so A for anaphase, apart. You want to pull them apart to opposite sides or poles. Okay, so they're still being pulled apart. And now cytokinesis is beginning here. Okay, so the same thing we see in binary fission, we see here where we start to invaginate. In telophase, everything that happened in prophase is going to be reversed. Okay, so we're going to get that nuclear membrane back. We're going to get rid of our spindles. They're going to fall apart. And then our chromosomes are eventually going to decondense. And now you're left with two cells that are identical to the cell you started with. And this is mitosis in a nutshell. So you have your prophase, prometaphase, metaphase, metaphase in the middle, anaphase, A apart. Okay, this is when they come apart. And then your telophase is your end. And cytokinesis that we talked about is just separating the cytoplasm. And so that's metaphase. Any questions, comments, or concerns about it?